Okay. All right.
Say go home and rest, don't travel to Florida, because they knew our plan was to deliver here.
Yeah.
South Carolina has no life flight. Like they will not let you out of state lines. They will not like they so we knew
We had to get there.
they were doing it. Are we ready? You guys need to go back and forth. That's fine. John hasn't even been introduced to Dr. Marc Arkovitz. So with that introduction, then you can introduce us again. Dr. Arkovitz, I introduced myself last night. I'm Julia Wynn. I'm the principal research coordinator for the DREAMS study, and I'm trained as a genetic counselor. So that's a master's degree, if people are wondering. And
Marc, I'm a pediatric surgeon, and I'm the clinical principal coordinator of clinical investigation. The DREAMS research study, which is based in Colombia, and is now working at the Seven Hospital, which is really awesome.
also, being honest, he's the brainchild of the study. Before we were funded by the National Institute of Health, he was the person running around and trying to recruit all these families and actually made us who we are today, or helped make us who we are today. So I'm just going to kind of go through a brief introduction of them. So I'm going to start off with, and I know that this is redundant for this room, but we'll start off with a very brief definition of the CDH, or congenital diaphragmatic hernia. And then I'm just going to go through some basic information about genes and chromosomes and DNA, because that's what I love as a genetic counselor, and it will help us understand why we're doing the DREAM study. Then we'll go over what we know about the genetics of congenital diaphragmatic hernia today, and then we'll introduce our study and talk about enrollment. So I took this lovely picture from the CHERUBS website. It's great, and I juxtaposed it next to an X-ray of a little girl with a left-sided congenital diaphragmatic hernia, and both of them are showing the telltale sign of congenital diaphragmatic hernia, which is you can't quite see it on the picture up there, but you can watch her nasogastric tube curl up and go into her chest, which is a sign that the stomach is present in the chest, which is not for every congenital diaphragmatic hernia, but for her. And then I labeled all of those other tubes that we see on these babies. And now I'm going to start off and go into Genetics 101. I'm going to breeze through very quickly from 8th grade science all the way into graduate level genetics. So stay with me. So we'll start off with just kind of getting into the picture of it all. And we all have billions of cells in our body. We all start off as that one cell. When the egg and the sperm come together, we make that one cell, and then those cells divide to make all the other parts of our body. And he's nodding along. You probably have this more recently than anybody else.
Just learned about this
So,
in school.
excellent. Ask him the questions at the end of the talk. So all of these cells generate all the tissues in our body. And inside nearly every single one of our cells is that structure there, and it's called the nucleus. And inside the nucleus is our genetic information. So all the cells of our body have a complete set of our genetic information. So the cells of our heart have a complete set of all of our genetic information, but just read the genetic information of our heart. The cells of our liver have a complete set, but just read the genetic information that's necessary to make the liver, and so on. And inside that nucleus, our genetic information is packaged on these structures called chromosomes. So there's 46 chromosomes that package all of our genetic information. And literally, if you stretch those chromosomes out, you get our DNA, which is what we all hear about in the news. And our DNA, in certain order, makes our 30,000 genes. So we have approximately 30,000 genes packaged on these chromosomes, shoved into nearly every single one of our cells in our body. And that's why we can take a blood sample, look at our genetic information, and from a blood sample, look at genetic information for all the different parts of our body. So we can look at genetic information for our heart, for our liver, just by taking a blood sample. So I'm going to actually go into and talk about how do we, as clinicians, study our genetic information in terms of for our families and trying to understand the genetics, what do we do to look at it? And I'm going to use an analogy that I like in order to try and explain the level of sensitivity that different genetic tests have, or what we can see from doing different genetic tests. So this first one is actually explaining that first. So chromosome analysis or a karyotype. And it's the same thing, we just use two different words to describe it. As with many things in science, we have multiple names for stuff. So if you think of our chromosomes as a set of encyclopedias, and so our chromosomes, remember from that last slide, are the structures that hold all of our genetic information. If you think of our chromosomes as a set of encyclopedias, a karyotype or a chromosome analysis is like looking to see that all the books are present. So we've made sure that there are no missing or extra books, but we haven't read all of the genes or the material inside. So we get kind of the basic overview. The other analogy that people sometimes use is if anybody's ever used Google Maps. No? Okay. So in Google Maps, you can look at the entire nation and you can make sure that all the states are present. And that's what a karyotype is like. And you are not zooming in and seeing the detail and making certain that all of the cities are there. So here we're looking to make sure that all the books are there. I have no idea what's inside those books. And on the right-hand side is a picture of our chromosomes. So there's 46 chromosomes there. I know that all the chromosomes are present, but I definitely haven't stretched them out so I can look at all 30,000 genes. And some of you may know in the room, there it says it's a 46XY. That's a male karyotype or chromosome analysis versus a female, which is 46XX. So the next step in a higher resolution of looking at our genetic information is doing what's called a DNA microarray or an array CDH, or all of these are essentially the same analysis. And what it is it's like taking each one of those books off the shelf and opening them up and making certain that each page is present. And so now I haven't read every single page, but I've made certain that I can see each page. And this picture here, how to put it over there. This picture here on the right is actually okay. So this picture here is like a drawing of one of the chromosomes. So you can see that it's got the bands. And then here's actually what the results of a DNA microarray looks like. And all these little squiggles here means that we have the right amount of material on our chromosome. And then this right here is showing that we don't have the right amount of material. See how it stretches? It's showing a deletion. So this test here, rather than making certain that all the chromosomes are present, it does that. And then additionally, it makes certain that there's no missing or extra pieces of the chromosome. So which are called microdeletions or microduplications. And then this single gene analysis or gene sequencing is essentially like taking those books off the shelf, opening them up, and reading each individual word. This is reading our genetic information or sequencing our genes. And this here is showing all the little squiggles that great people can interpret them to know a lot. Anyway. Interpret to know whether or not there's missing or extra words or letters in our genetic information. Okay. So this is just showing a couple different things that you can detect by doing these. So this is a chromosome analysis. You can see that there's an extra chromosome in this karyotype. So there's 47 chromosomes rather than 46 chromosomes. And this is showing a common genetic condition called Down syndrome.